So this is chapter 27, and chapter 27 is going to be about bacteria. Now, when we think about bacteria, we usually think, ew, disgusting, it's going to harm us, and there's definitely bacteria out there that'll do that. But keep in mind that a lot of bacteria that are out there are actually beneficial or even benign, which just means that they're there, but they don't do anything to hurt us. So very, very, very large group that we're going to be talking about. And there's two groups that we are going to kind of branch these into. So you've got the regular bacteria, and then you have the archaeobacteria. So archaeobacteria are going to be ones that live in like really extreme environments, like under the ice or in anoxic conditions or something like that. Whereas regular bacteria is going to be, living in every single type of environment you can think of. Okay, so we'll get more into the archaeobacteria in a little bit. So let's talk about the form that they can have. So remember, these are prokaryotes, which means that they don't have any organelles. Now, as far as their form, they can have one of these three types. I'm going to make this smaller again. So you can see they can be coccus, which is going to be little circles. You can have bacillus, which are going to look like little tic-tacs. And then you can have spirillum, which is going to have that spiral shape. So those are the three basic shapes that bacteria can have. And the coccus and the bacillus tend to form colonies, like the bacillus will join kind of end to end and form chains, whereas the spirillum tends to kind of sit by itself. Okay, so there are going to be some types of bacteria that can release spores, and spores are like mini bacteria that can grow into new ones, and that's the way they can asexually reproduce. So if you remember a couple of years ago, they were talking about anthrax and freaking out about it because people were sending envelopes that had the spores in there. So that's a perfect example of spores that were in the media. All right. Now, if we are going to compare prokaryotic versus eukaryotic cells, you've actually done this before. One thing that they're all going to have in common is that prokaryotes are single-celled. So even when they form those colonies that we were just talking about, they still retain their own individuality. Whereas with eukaryotic cells, you can have a whole bunch of cells that are actually forming an organism, and they can't really live by themselves, right? So I don't know why that always comes up. Okay, next thing is the size. Now, prokaryotes are going to be smaller than eukaryotes, and that makes sense because prokaryotes don't have any organelles or anything like that. Another difference is going to be the way that their DNA is set up. So in prokaryotic DNA, it's going to be set up circular, and eukaryotic DNA, it's going to be linear. The next one you may remember from Bio 111, cell division. So prokaryotes are going to divide using binary fission, which is very simple, where they just copy their DNA. One goes to one side, one goes to the other, and the cell splits. Eukaryotic cells are going to divide using mitosis, right? So they're going to go through prophase, metaphase, and all those different phases. The next one is one that we already talked about, and that's that prokaryotes don't have organelles. They do have ribosomes, but that's pretty much it. No nucleus or any of those other ones. And then this next one, a flagella, that's something that both prokaryotes or eukaryotes can have, but prokaryotes are going to have a flagellum that rotates like a boat propeller, whereas eukaryotic cells is going to have one that kind of goes back and forth. It's a little bit more advanced. Oh, this thing, okay. There we go. All right. The next one, the last one, is metabolic diversity. So this is kind of interesting, and that's just basically that prokaryotes can do a lot of different ways of getting their energy and getting their carbon sources that eukaryotes cannot. So we'll get into that a little bit later. All right, let's get into prokaryotic structure. So one thing that we've talked about a little bit is that prokaryotes are going to have a cell wall. And in these pictures, it's going to be the white that you see here. So you can see in this picture, I'm showing you gram-positive bacteria. And gram-positive bacteria is going to have a very thick, very simple cell wall. Now, if you compare that to gram-negative bacteria, you can see the white part is very thin, right? And so gram-negative bacteria is going to have a thin cell wall, but the whole outer layer is a lot more complex. So we've talked about gram-positive and gram-negative bacteria as far as colors, too. So if you remember, gram-positive bacteria is going to turn purple with the gram staining technique, and gram-negative bacteria is going to look pink. And this is a great picture of a slide because this shows all the different forms they can have. So you can see there's some coccus right here. You can see here's the bacillus, and then you've got a couple of sporillum, right? So those are those three shapes. So any of these shapes can either be gram-positive or gram-negative. All right. So as far as examples of these, as far as gram-positive bacteria, examples would be anthrax, botulinum, which causes botulism, staph, and strep. Those would all be examples of gram-positive bacteria. As far as gram-negative bacteria, E. coli, the bacteria that causes typhoid, cholera, meningitis, and gonorrhea would be examples of gram-negative. In general, gram-negative bacteria tends to be a little harder to kill with antibiotics, and gram-positive tends to be a little easier. And what that means is there's more of a range that can kill the gram-positive, whereas with gram-negative, you usually have more specific ones. And one of the reasons for that is because gram-negative has that complex cell wall. So if we look at this picture again, gram-positive bacteria, that cell wall is very simple. It's thick, but it's simple. And so what can happen is the antibiotic can get into the bacteria really easily. Whereas with gram-negative bacteria, you can see that it's got a lot of things going on, and these little green things that you see here, those lipopolysaccharides, those are actually going to sometimes secrete toxins and things. So tends to be a little bit scarier and sometimes a little harder to kill with antibiotics. Alrighty. And the reason that we can take antibiotics without hurting ourselves is because we don't have a cell wall. And a lot of times that's what they're going after, right? Another thing that prokaryotes can have, this is a good picture to show you. First of all, what you want to notice is that this prokaryotic cell has DNA, but doesn't have it contained in a nucleus. The other thing, sorry, is that they have these little hair-like structures sticking off, and those are called pili. And what those do is those allow them to meet up with each other, and they can actually do conjugation, which is where they exchange genetic information, or they can also attach to stuff. So let's say this is some nice bacteria that wants to grow in the back of your throat and cause strep. They can hang on to the back of your throat with these and start to eat away at your tissue, right? So that's going to be another thing. Now this last one here that is specific to these guys is they can form what are called endospores. And endospores is what you see here. And usually they'll do this when they get exposed to an environment where it's very harsh and it's probably going to kill them. And what they could do is just kind of make this shell, this protective covering, over their genome so that they can sit there until environmental conditions get better. That could be hundreds of years. So it's kind of crazy that they can do that. Sometimes it's just a couple of days. Other times it could be hundreds of years. So those are all going to be characteristics of prokaryotes. And in this next part, we're going to get into their nutritional diversity.